Good morning, student. Today I welcome all the student in biology class of standard 10. In this class, we are going to discuss the constituent or the raw materials that is required for the process of photosynthesis. In the previous video, we have discussed regarding the autotrophic nutrition or autotrophic type of nutrition, how the plants prepare their own food. In the process of photosynthesis, we have discussed that plant needs carbon dioxide, water, sunlight and chlorophyll. Today we will discuss about the necessity of sunlight, chlorophyll and carbon dioxide. Sunlight, chlorophyll and carbon dioxide. For what purpose? For the formation or making of starch. We would test in the preparation of a starch. These things are vital things. How would we prepare? First we will talk about the sunlight. And if we are talking about the sunlight, first we take a potted plant. Potted plant जो होना चाहिए आपका, इसमें suppose we take a leaf, maybe many leaves, we have taken. I have not drawn the leaf and other things. A long leaf type plant is required. Potted plants, so that we can easily put it into the dark room as well as the sunlight. We cannot do the same activity using a big tree, because big tree cannot be uprooted and kept into the dark room. First, these potted plants has been kept into the dark room for 36 hours. Maybe more than it. It may go for 3 days also. 36 hours to 3 days. 36 hours to 3 days. We keep it. Why we keep it so long for almost for the 3 days? We keep it for the 3 days in which we consume that the starch that is available in the plant has been consumed up. उसको plant को consume कर लिया है. After this one, after keeping it for the 3 days in the dark room, and why we are keeping it For the 3 days in the dark room, we assume that all the starch that is present in the leaf had been consumed. Glucose is instantly used by the plant for the release of energy, and the rest of food is stored in the form of a starch, that is starch. When we are keeping it for the 3 days in the dark room, after that we just put a aluminum foil on it. After keeping the aluminum foil on it, we keep the potted plant into the sunlight. This is the aluminum foil. Why aluminum foil we have wrapped it? We have wrapped the aluminum foil in order to protect it from the sunlight. Then we dip this potted plant and keep in the sunlight for 6 to 8 hours for the starting of the process of photosynthesis. As soon as it has been kept for 6 to 8 hours, photosynthesis has been started. Leaf has prepared the photosynthesis of glucose. The rest amount has been kept or stored in the form of a starch. We pluck this leaf. After plucking this leaf, we after plucking this leaf, which has the here aluminum foil on it, we boil it in water. Where we boil it? Boil it in water. Now question comes, why do we boil it? And if we boil this leaf in the water, what happens? We have removed the aluminum foil. When we boil this leaf in water, then the leaf becomes dead. It is killed. And cuticle that is found on the surface of the leaf, the upper surface of the leaves, it gets loosened. Cuticle, what? Cuticle and cell wall get loosened, and when it gets loosened, it permits two things. First thing, it allows the chlorophyll to come out when we boil it in the alcohol, and it also allows the iodine solution to enter into it. We will discuss later on. First, we have boiled it in water. When we boil it in water, it has been assumed that the cell is killed, cell is dead, their cuticle and cell wall has get loosened. Then we boil it in alcohol indirectly. Indirectly, what does the indirectly mean? It means in this we keep the water, then we keep a test tube. This test tube is filled with or ethyl alcohol. Why? Why we boil it indirectly? If we boil alcohol directly, then it would catch the fire. We have kept the leaf into it. leaf has been kept into it, then what happens? The first when we had boiled the leaf into the water, it has been dead. The cuticle and the cell wall is killed. It becomes the lucerne, and when we boil it indirectly in the alcohol, then it is assumed that its chlorophyll has been taken out. Now the leaf becomes colorless. Leaf has become colorless, and now as soon as it has become colorless, we take it out, we wash it clearly so that no chlorophyll remain stick to it, and after washing it we place the glass slab and we put the iodine solution. Two things that we have to keep in mind over here when we will do the other activities, that iodine solution is not the pure iodine solution. Actually, it is a potassium iodide or potassium Ki3. It is actually Ki3 solution because iodine is not soluble or very less soluble in water. It is also not soluble in non-polar solvent. To make it solvent, we add it to the potassium iodide. So we can say it is a mixture has been prepared. It is potassium triiodide. Triiodide solution. It is a triiodide solution is there. This triiodide solution is used to detect the presence of starch. Then what happens? In the starch there are two types of molecules are present. One is amylose and amylopectins. It is around 25% and rest 75% is occupied by it. It is long chain. It is made of long chain. It is of branch chain. This is long chain. This is a branch chain. Only the presence of amylose, that is the molecules of starch, molecules of natural starch that is found in the plant. The two types of starch that is found, one is that is the amylose, which is of long chain. Another one is the branched one, amylopectins. Only this amylose gives the dark blue color because the potassium triiodide solution, Ki3 solution, gets embedded into this long chain of amylose and it absorbs the most of the light that is falling on it. Since it absorbs most light falling on it, so it looks blue, dark blue in colors, and potassium triiodide solution's color is brown in color. When it is in the concentrate form, high concentration form, it is brown in color. When it is a less concentration form, it is yellow in color. You have to keep in mind, when it is less in concentration, then it is yellow in color, but when it is high concentration, then it is brown in color, and we use the triiodide solution instead of iodine solutions. But since iodine percentage is more, so we say the iodine. Starch is an indicator. We can say in another language, starch is an indicator which turns iodide solution, triode solution into blue black color. When we put it, we find that the parts which was covered over here, it has remained the same color, colorless, but these parts had turned dark blue. This shows that these parts has the starch present in it. It has the starch, and here is no starch. It means up to this place no photosynthesis had taken place, no photosynthesis had taken place, because this part of the leaf has not received the sunlight. This activity shows that we require, means plant, not we, plant required sunlight for the process of photosynthesis. फोटोसिंथेसिस के लिए उनको क्या चाहिए? सनलाइट चाहिए. हमने कैसे प्रूफ किया? हमने देखा एक एक्टिविटी के द्वारा that they require the sunlight. Then another things that we want to show that chlorophyll is also an essential part of photosynthesis. The activities remain the same, but one thing changes. What are the things changes? Instead of a single plant, we take a plant which has the two colors. One is green, one is non-green color. We take the example of money plant. The money plant has the two colors. One leaf has the two parts, two colors. One is green, another is non-green. We can say it is a light yellow in color. It means if we imagine, if we say that the only green part perform the photosynthesis, other parts do not perform the photosynthesis, we have to show it, because green parts contains the chlorophyll. The same activity we'll do. We take this money plant for 3 days inside a dark room, and when it has been kept inside the dark room, we assume that its starch that has been present in the chlorophyll has been present in the leaf has been consumed or eaten up. Now we keep it into the sunlight for 6 to 8 hours. The same activity we have done in the case of when we were testing that the sunlight is necessary. The same thing we would do in this case also. We take the potted plants where in the dark room for how much? For the 3 days. And why we keep that 3 days? Because we think that the starch that is present into it has been consumed up, because in these 3 days, in the absence of light, no photosynthesis activity is taking place. Now we keep this plant into the sunlight for 6 to 8 hours. Now we pluck the leaf. Before boiling it, we wash it and make a sketch drawing of it, so that it can remember us to which part were the green and which part were not green, which part were green and which part were not green. And then we boil it into the water. After boiling the water, and what happens when we boil in the water? Boiling the water means it kills the leaf. It softens the cuticles and the cell wall. Then we boil it into the alcohol indirectly. And when we boil it into the alcohol, all its chlorophyll has been taken out. It becomes colorless. We wash it properly so that no pigment of chlorophyll remain stick to it. Now the same activity, we just drop triiodide solution onto it, means iodine solution into it, and the color or the part of the leaf which were green in color has turned blue black, and the part which were non green in color color has not turned blue black. This activity shows that green pigment, that is chlorophyll, it is necessary for the process of photosynthesis. If we see the plants like which has the leaf which has the two colors, it means the color which is green perform the process of photosynthesis, otherwise they are not performing the process of photosynthesis. It means we have discussed the two things. First, sunlight is necessary, then chlorophyll is necessary. Third things that we do that carbon dioxide is necessary. For the carbon dioxide is necessary, we take the two potted plants, identical potted plants, same type, we may take it from the nursery itself, identical plants, same plants, same height, everything is remain the same. Then both the plant which has leaves, many leaves, we just keep it on the glass slab. We keep it on the glass slab and cover it with the watch glass. Glass slab में रख दिया. Two potted plants, we can do the same. We also cover it with the watch glass, and the joint we have just try to seal it with any thick substance. We may use the Vaseline, other things. In one plant, we do potassium hydroxide. Another one we have not keeping anything else. First, before keeping the potassium hydroxide, we have taken this plant and kept into the dark room for 3 days. The same thing, same conditions. What we think that during this period, whatever the starch is present into it, it has been consumed up. Then we keep it in the sunlight, making this apparatus, covering it with the watch glass. In one, we keep the potassium hydroxide. Potassium hydroxide is a strong alkali, and carbon dioxide, it is non-metallic oxide which is acidic in nature, and acid and base reacts, it forms salt and water. It means it reacts vigorously, and all the carbon dioxide, what amount of carbon dioxide that is present into it, it has been taken it by the potassium hydroxide. It means here is no carbon dioxide. It means here no CO2, yes CO2. In one watch glass carbon dioxide है ही नहीं, एक में carbon dioxide है. We have kept it for 6 to 8 hours in the sunlight. We remove the watch glass, plug the leaf from each other's, marking it from where we have taken the leaf, part 1 and part 2, part 1 and part 2. We may use the thread to identify. In one, we may use the thread, tie it up so that it is from this one, it may be without thread. The same activity, what we do, we would boil it, then we boil it indirectly into the alcohol, keep in mind, but we cannot boil it directly, because if we try to boil it directly in alcohol, it would catch the fire. After boiling it into alcohol, it becomes colorless. Why it is becoming the colorless? Because all its alcohol has been taken out. Now we wash it, put it on the glass slab, and then we start pouring the iodine solution onto it. The leaf which was present and we had taken and plucked from this pot has not turned blue-black, but the leaf from this pot has turned the blue-black. It means here the photosynthesis has taken place. Here photosynthesis has not taken place. Why the photosynthesis has not taken place? What is absent over here? Here carbon dioxide is absent. Here carbon dioxide is present. Because it is sunlight, watch glass, sunlight is available to them. Sunlight तो मिल ही रही है. Chlorophyll है present. What is not present in this one? Carbon dioxide is not present. So from this activity, at least we are able to understand, we are able to conclude that sunlight is necessary, that chlorophyll is necessary, that carbon dioxide is necessary for what? For the process of photosynthesis. What the second thing we have learned? What happens when we boil the leaf? When we boil the leaf, it is dead. Cuticle, that is the upper waxy surface, becomes loosened, and cell wall also become loosened. What is benefit when it becomes loosened? When we boil it in alcohol, it allows the chlorophyll to come out, and when chlorophyll has been taken out, it becomes colorless. We wash it, and why we wash it? Because no any chlorophyll pigment should be attached to it. Then we put the iodine solution, iodine triiodate solution, means iodine solutions, and the same conditions when it has allowed to come out the chlorophyll, it also permit the iodine to go into it. The next thing, what important things that we have understood? We have understood that the starch is of natural of two molecules, made of two molecules. One is amylose. Amylose is almost 25%, 1/4, and amylopectin, it is 75%. It is the long chain, it is the branch chain only due to it, because in this long chain the iodine solution get embedded into it and make the structure which absorbs most of the light, and when it absorbs most of the light, reflect less light, then it means it is seen dark in colors. So blue, dark blue color is appeared. This is not actually allowing the iodine to enter into it, because it is so complex branched chain. Yes, a little amount of iodine penetrate into it, but it gives very light color, yellow color. So we have got it, what are necessary for the process of photosynthesis. Go through this video, this activity, and try to understand what we have discussed. In the next video, we will do the another part of this. Thank you.